Hopefully it should work. Okay, so how did we feel? So raise your hand if we've already had discussion. Cool. How are we feeling post first discussion on material? Because you got to work on a little bit of problem set. Anyone? Don't really care. Okay, how about we do like a little thumbs up? Thumbs up if you're feeling good about first topic. Middle? Anyone middle right now? Anyone scared from how we're all talking about this class? Okay. I forget who and somebody in here. Yeah, was it you? You're the mechie guy taking this as a gen ed. So sorry. Like, so sorry for that. Okay, let's just get started. Should I start and then you can do the
We can rotate every other slide.
Okay, so fun high school bio type stuff. Prokaryotes versus eukaryotes. So prokaryotes are the simple stuff. Eukaryotes are more complex. If you heard the word nucleoid, and you're like, isn't that the same thing as nucleus? Nucleoid is this little tiny thing right here, and it's made out of like this spaghetti looking thing called what's it, chromatid, something like that. Don't worry too much about the distinction in those. Main thing you want to know, prokaryotes lack those internal membrane-bound organelles. It has the nucleoid, not the nucleus. And prokaryotes include the bacteria and the other one. What is it? How do I say it? Archaea? Archaea. Yeah, don't really want to go too into the.
Does anyone know an example of a type of membrane-bound organelle that a eukaryote would have that a prokaryote wouldn't have? Any membrane-bound organelle.
Like the meme one?
Exactly. Endoplasmic reticulum is one of them. So that'll be right here, all this stuff. I can see that right here. Then there's mitochondria. Then there's the nucleus. Basically, so just like the name says, those membrane-bound organelles are just organelles, parts inside the cell that have a membrane around them. That's what the prokaryotes don't have. All right. And then some things both of them share in common is they both have DNA, which is important, obviously, to replicate and pass on genes to offspring. And then they both have cell membranes. We'll talk about that a little bit more later, but it's basically a phospholipid bilayer, which means that there's two layers of phospholipids on top of each other. And those will just be like the wall to the whole cell. And then both of them can have cell walls. So that doesn't mean they always have cell walls, but they can. For example, in eukaryotic cells, plant cells will have cell walls, but animal cells don't always have cell walls. Does that make sense to everyone?
Okay, so viruses. And we're going to talk about a question later on that's like kind of related to something on your problem set that asks about viruses. What is a virus? Genetic material is a protein shell. It's pretty small. Special thing about viruses is that they need a living host to replicate because they don't have the required tools, which is, we'll talk about the specifics of that in a bit. And they have their own metabolism. So it's interesting because it can't really do its job unless it's like linked to something that's giving them energy. Hint, hint to a question that we'll talk about there.
So it's important just think of these things as DNA wrapped in something and they're little parasites that if they're on their own, they can't replicate, they can't pass on any of their information. So they're always looking for a host because the host will have, like we talk about later, the ribosomes, the other organelles that they need to replicate.
Okay. Yeah. So let's just do the first bullet point. Like let's just see what people are saying. We're gonna go through these one by one. So to be considered a cell, you need three things. The first thing is you need to be able to what? It's a very simple term. Does anyone know what it could be? Any guesses? Reproduce? No, but we're kind of on the right track. No, think simpler. Think simpler. No, no. Super, super simple. It's actually
it's gonna say all.
Okay, yeah, I can't tell you. Okay. Um, okay, we'll just go through these one by one and then I'll show you guys because I don't want to show it all at once. Okay, so there's the first one, there's the second one, which is needs to be able to carry out something. I think one of you already said it. It's a process. No, what do we do when we break things down? What is that called? Guesses.
You said it.
Yes.
Metabolism. Yeah.
Yep. So it needs to be able to carry out metabolism. That's the second one. And the third one, it needs to have a blank. Think about viruses. In order for viruses to carry out their job, what do they need? They need to like latch onto something, right? What can we call that? Organelles? No. Again, think simpler. A host, what does the host have? Energy. It needs to have an energy source. So let's go through all of these. First ones need to be able to grow. Again, think super simple, right? This is everything that to be considered a cell, you just need these three basic things. So think simple. You need to be able to grow, you need to be able to carry out metabolism, and you need an energy source. That's really important. Otherwise, how are you supposed to do your job as a cell?
Does anyone have any questions on those? Those can get a little confusing sometimes. All right. So now with what we know about living things and what we know about viruses, do we think viruses are living or non-living? Non-living, why? Okay. Does anyone else have any other opinions?
They can't make their own energy.
Okay. Okay. Good. So they're in between. So I've seen more often than not them be considered non-living just because the important factor that they can't reproduce on their own, but they do still have some traits that would make them living. So scientists consider them in between, because they can replicate if they have the help of another organism, and they do have the genetic material and all that stuff.
Yeah, like the whole thing about viruses is like you need a host. Like they can do these things, but they need to be able to like attach the host first before it does that. So it doesn't have its own energy source. It doesn't have its own things. It needs to attach something to be able to do that. So that's why it's kind of in the in-between because it can do these things, but it needs something else to be able to do those things sometimes. Does that make sense?
Okay. Transcription. Does anyone have an idea of what transcription is? Exactly. So first, when you have a cell, right, you have the DNA in the nucleus. At least in a eukaryote, it's in the nucleus. And then the end goal, the purpose of that DNA is to make proteins. The DNA is like the instructions, like the cookbook for what you're trying to cook, right? The proteins at the end. What you need to do first is you need to translate it into something that you can understand. So for example, say you have that cookbook and it's in some other language, you can't understand it. First, you need to translate it or transcribe it into something that you can read. So that thing that we can read is the RNA, specifically mRNA, right? Basically what happens, well, I'll show you guys a picture in the next slide, but we're gonna have something called RNA polymerase, moves across the DNA, and it makes like a replicate strand of that in RNA form. And then that RNA will eventually be used in translation to make the proteins. So since it's using the DNA, it would happen in the nucleus in eukaryotes because the nucleus is where the DNA is found. But since prokaryotes don't have those membrane-bound organelles, there's no nucleus to store that DNA. So that would just happen in the cytoplasm or just the main body part of the cell. And then here's an image of what I was talking about. So pretend all of this is in the nucleus, and we have the template strand right here. This is the DNA in blue, right? You're getting one part of it, and we call the part that's being transcribed the template strand. And you have this big thing right here, which is the RNA polymerase. It's a protein that basically reads the DNA. As it's moving along the DNA this way, it's coming up with a complementary base of RNA. So for the base T right here, it's making an A. Again, here, same thing. Then for a G, it's making a C. And this mRNA is going to be used as the new recipe from the cookbook that you can understand and make the protein out of it. Does that make sense to everyone?
Does anyone know the difference in bases between RNA and DNA?
DNA has a double base while RNA has.
So I was looking for something simpler, right? DNA, whenever I look at it, it has T. You guys see like the tag TTT? RNA has U instead of the T. Okay, so that's kind of big because on the exam, you're going to be asked to like translate into different things. RNA has the U, DNA has the T. Just something to keep in Okay, so we have mRNA, and there's different types of RNA, which we'll go over at a later time, but we're keeping it simple. So just like Yago said, step one of this recipe is we need to change DNA to mRNA so we can read it. So we have that, right? And what translates that from DNA to RNA? What is that thing called? RNA what? Polymerase. RNA polymerase is what does that job. Okay, so it goes along and it's making it from DNA, it's giving it to RNA because we need to read it. We have that. Now we have this mRNA string is what we end up with. We need to go from this string of random bases, essentially, into a functional protein. So there's a couple steps here. So translation, I just want you to think of, it goes from mRNA to an amino acid sequence. We'll talk about what happens after, but that's like the arrow that I want you to think about. Amino acids. We'll talk about the differences between them. We'll talk about the structure and stuff later on, kind of what you did in your discussion. But we're just translating that mRNA into actual useful amino acids. And when you put a bunch of amino acids together, you make what? You make a protein. Okay, so are we getting the steps? Like step one, DNA to mRNA, we need to understand it. Now we're going from mRNA to the actual amino acids, which is making up this protein. Does that make sense? Okay. So amino acid sequence form. So amino acids, they each make up like 3 base pairs. Just a little detail. So you have 3, 3, 3. These are all amino acids being strung together because we're making a what? What are we making? Protein. Making a protein. Based on that chemical structure. So based on whatever amino acid is going where in this string, like we're making like little bead strings. Based on whatever is where, it's going to fold a certain way. It's going to fold a certain way. So if we have a certain amino acid here followed by a different one, if we switch those 2 positions, it's going to fold differently. Okay. And that structure determines what that protein is going to do. And we call this, we say structure determines function. Okay. So keep that in mind. Does that make sense? So the chemistry is changing. When the chemistry changes, the fold of everything changes. And based off of that, the job of that protein changes. Does that make sense to everybody? Okay. So step 1 is DNA to what? mRNA. Second step is mRNA to what? Amino acid sequence. Amino acid sequences, but what do amino acids make up a what? Protein. So, and based off of that, the structure determines the what? Function. Okay, so that's the key that you're going to have to know. Structure determines function. It's going to come up this time. It's going to come up in later units. Whenever you're changing the chemistry of something or changing the shape of something, as a result, you're changing the job of whatever that thing is doing. Cool? Okay.
And then this will take place in the cytosol in both eukaryotes and prokaryotes. So remember, in eukaryotes, the step before transcription happened in the nucleus because that's where the DNA was. So now the mRNA was made from that last step. That mRNA has to be brought outside of the nucleus to the ribosome for translation to happen. In the prokaryote, since it didn't happen in the nucleus to start with because there is no nucleus, it just happens basically right next to where it was in the cytoplasm again.
Because the prokaryotes don't have a what? Nucleus, right?
And here's the image of translation. So we have the code, the mRNA code we made from the last step, and we have this ribosome right here. The ribosome will be moving around along this strand, and you have tRNA comes in, and if you see each tRNA has a 3 base codon. That 3 base codon is important because it's specific for a certain amino acid. So this little ball right here will be the amino acid, and it'll bind to the matching sequence. Once it binds to the right sequence, it'll attach that amino acid specific to it to the growing amino acid chain, and that amino acid chain will become the protein. And then this is basically the whole process summed up, like Anjali was saying. So we start with the DNA right here. The DNA template strand is turned into an RNA, matching RNA strand. And then that RNA is read into different amino acids that make up the chain. So cookbook in a language we don't understand. Cookbook is being changed to a language we understand. This is the language we understand. And now we're making the meal or recipe with it. Make sense? And then this is showing in a eukaryote, it shows how the transcription happens in the nucleus. And as soon as you have that mRNA made, you have to bring it outside the nucleus to the ribosome where translation happens. And we finish with that protein right there. Make sense to everyone?
Questions? So what is step 1 called? What do we call that process? Transcription, and then the rest of it is what? Translation. Okay, so based off of what Yago just said, why can prokaryotes do transcription and translation at the same time, but eukaryotes can't? Yes, they don't have a nucleus. Yes, that's essentially what it is. But what is separating those processes? What's separating them? Yeah, space and time, right? So eukaryotes, transcription and translation are separate in space and time. So transcription happens where in eukaryotes?
In eukaryotes or prokaryotes? What they say?
In eukaryotes.
Yeah, in eukaryotes, it's in the nucleus. Remember, DNA is in the nucleus. In prokaryotes, there is no nucleus because they don't have membrane-bound organelles. That's one of the major differences between them. And so it just happens in the cytosol. Can anyone think of why it'll be important that in prokaryotes, translation and transcription can happen at the same time, but not in eukaryotes? Can anyone think about what problems that might cause in prokaryotes if they're just happening same time? So one problem in eukaryotes, you'll see that these processes aren't perfect. So there will be mistakes in transcription and in translation. They're rare. So usually most of the time the cells can fix them on their own. But for example, in eukaryotes, if there's an error in transcription, it's still in the nucleus and it has time to kind of fix it before it gets brought out to be translated. Because you don't want to translate the wrong thing and then put in the wrong protein, and the wrong order could just screw up a bunch of stuff. So it has time to fix that in eukaryotes. But if you're in prokaryotes and it's happening same time, it's much harder to control those mistakes because you can make the wrong copy of something and then it be translated right away. Does that make sense to everyone? So that's why it's important to know that they occur at the same time in prokaryotes and not in eukaryotes. You can.
Okay, so gene expression consists of transcription and translation. And what does transcription? What is that thing called? RNA polymerase. Okay, I think that's probably your question set. RNA polymerase is responsible for changing DNA to what? What is that called? What? mRNA. Okay, that's what it does. Translation, we already talked about mRNA to amino acids, right? A bunch of amino acids make up a what? Protein, right? And a bunch of stuff depends on that. So, this is the question that I really want to ask you. So let's go over those steps. I want to make sure we have these steps down because it's easy to get mixed up. So here's your little word bank. You can use multiple words for different questions. Let's start with the first one. The blank sequence determines the blank sequence. Who knows the answer to this one? Yes, DNA and RNA. Step 1. The DNA sequence determines the RNA sequence. Okay. The blank sequence determines the amino acid sequence. RNA, right? What specifically, which RNA? M. We're going to talk about the different ones in like way later.
And
but
what step is that? Translation.
right? So step 1 is which one?
Transcription.
Transcription. That's just the DNA to mRNA. Second, and everything else, translation. Okay, amino acid sequence determines protein blank, aka the blank. Who knows this one? Yes. So, amino acid sequence determines the protein folding. Why? Because the chemicals, right? You put certain chemicals, certain places, it's going to affect the folding of this protein, which determines the structure of this folding. Protein folding, that's just a fun name for like how it's structured and looks. And meaning the blank determines blank. What is that phrase? Structure determines function. Okay. So do we all have those steps down? How are you feeling so far? Good. Thumbs up.
Question right here.
Yeah. There you go. Yeah. Yeah. So whenever you hear like when you're going on the exam, she loves mutation questions. Okay. So basically for any step oriented like unit that we're doing, you need to think about it in terms of like, okay, I know step 1, I know step 2, because all of the questions are going to be if this step gets interrupted or if something happens to this step, what happens to the overall process, right? So that's really important. You're going to get a lot of mutation questions on your exam. So when we get into like the actual coding and like how we translate things, it's going to go, okay, what happens if there's a mutation here? And like, what's the end result going to look like? Because that's actually applicable to us, right? That happens to all of us. And that's that simple reason is behind a lot of like really tough medical things. Yeah, so just keep that in mind for your exam. There's gonna be a lot of mutation stuff. Okay, differential gene expression. Differential gene expression. It sounds like a fancy term. I promise it's not as fancy as it sounds. I'm going to tell you what it means in like under 10 seconds. Okay, you ready? All of our bodies contain the same stuff and they express different things. So everyone in here, we all have the same genome. We all have the same like gene library. Okay, we are all made up of the same stuff. But none of us here look the same. None of us here act the same. None of us have traits that are really identical to each other. And it's because our bodies express different genes. So cells are made of the same stuff, depends on which of those genes are expressed. So that expression is what makes us different from each other and what makes cells different from each other. So like precursor red cells make hemoglobin and B cells make antibodies. I don't love that example, but that's essentially what that phrase means. She's going to say differential gene expression a lot. Okay. So I think there's a question or problem set that's like, here's different types of cells, or they all have the same genome, but why are they different? And it's because they're expressing different things. And this could be based on because of the environment, right? If you have traits, maybe you're more prone to expressing that trait in a certain environment, right? So just overall, we're all made of the same stuff, but we all express different parts of those genes. And depending on whatever you express, that's what makes things different from each other. Any questions on that?
So has anyone heard people say that I don't know the exact numbers, but say like we share 97% of the same DNA as ants or something like that. Like that's basically what this point is saying, that a lot of organisms are the same, have the same genome, same genotype. Everyone knows what genotype means. Basically like the DNA that we're made up of. But the reason our phenotype is different, our physical appearance is different, is because those genes are expressed differently. Does that make sense? Okay, so fill in the blank. The point of differential gene expression is to show that differing cells have the same blank, but different blank. Anyone have any idea? What do we all have the same? Genotypes. And different expression of those genotypes or genes creates a different what? Phenotype. So physical appearance. Does that make sense? Okay, so now amino acids. Like we talked about, amino acids are the building blocks of proteins. You'll have a bunch of amino acids put together in chains, and that makes the protein. Each amino acid has the same basic overall structure. They all have an amino group. I remember this because at the end in amino, and amino groups always have a nitrogen, and the nitrogen will be bound to hydrogens. And then we have a carbon that connects that amino group to carboxyl group. And I remember that because the C or the car for carbon and the oxyl for the oxygen. And then there's a hydrogen connected to that middle C. So this part that I just talked about, those three things, same for all amino acids. What makes the amino acids different is this R group right here. So connected to that central carbon, each amino acid will have a different variant right here. It can be as simple as having another hydrogen there. That'd be glycine. Or it could have a whole ring. Make sense to everyone? And if you see, the amino group will have a positive charge and the carboxyl group will have a negative charge. Unless this R group is charged, the overall charge of amino acids will be neutral because they cancel each other out. But then again, there's the charged R groups that could, if the R group is negatively charged, it'll have an overall negative charge because these cancel out and it'll be left with just the negative. And if it has an overall positive charge on the R group, these two will cancel out, just leaving the positive charge on the R group.
Where they're
Yeah, I go back. Okay. And basically, so they'll hook up to each other by amino groups and carboxyl groups joining together. So literally just think about a lot of these together and you just keep adding ones next to each other, and they'll make bonds to each other. Anyone know the types of bonds? What we call that bond? It'll be peptide bonds. All right. Some definitions you guys will need to know. A peptide is just a chain of amino acids, so a short string. And then if you have a polypeptide, poly, think more, longer string of amino acids. Polymer are long chains strung together, made by repeating units. So like RNA polymers, DNA polymers. And then protein. Protein can be interchangeably used with peptide or polypeptide. A protein will just be the string of amino acids.
Yeah, there was like a question for your discussion. I remember that was like, which one is the polymer? Like, which ones are polymers? Polymer just means it's just a long thing that's made up of the same thing. So like RNA polymer is made out of what? RNA. Amino acid polymer or a peptide, they mean the same thing. Okay, so don't get these confused. Polymer just means that you're making a thing with a bunch of repeating steps. Okay. So okay, he just said this. So what makes amino acids different from each other? We know that 3 out of 4 of those structures are pretty much identical on every single amino acid. What do we call that other part that makes each of them different? R group. R group is the variant. R group is how we know amino acids are different from each other.
And another name for the R group will be side chain. Side chain and R group are the same thing. This part right here, like we talked about. Okay, so now nucleotide structure. What are nucleotides used for? Who knows what we use nucleotides for? Exactly. So like amino acids that all together make up a protein, nucleotides all together make up DNA or RNA. All right, so the basic structure of these, they all have a phosphate group right here. The phosphate will just be the phosphorus with oxygens around it. You'll have a pentose sugar. That's this right here. And those two are the same on every single one. It'll be different. The sugar will be different on DNA and RNA by just a OH group, but that's not that important for you guys to know. But that's just why DNA is deoxyribonucleic acid and RNA is just ribonucleic acid. So those two are the same in all of them, just like the amino acids have the same backbone. But then the difference, or the R group in these, would be the bases. So like we talked about before, the matching bases. So we'll have the adenine, or the A, thymine, the T, guanine, the G, cytosine, the C, and then in RNA, uracil, the U. So as you can see, they're kind of similar to each other. They have the same basic structure, but they have a little bit of differences. And depending what base is bound to that pentose sugar is the nucleotide you have in that DNA or RNA. And how these stack up onto each other, you'll have the phosphate group will make a bond to the sugar of the next one. So if you add one on top of here, you'll make a bond between the sugar and the phosphate right here. Does that make sense? And we call that a phosphodiester bond.
Let me say what this one's like DNA. Like this one.
I think this would be DNA.
I think it's fun.
No, yeah. And so this one right here would be RNA because it has the OH group right here.
We had a TA say earlier today, I thought this was a really great little trick to know. So DNA stands for like deoxyribose, right? And then RNA is just ribose. So when you think deoxy, when you're talking about DNA, we think like delete oxygen. Think delete oxygen. Okay. We know this is RNA because it has that extra oxygen guy there. But just a little memory trick when you're thinking about the structure and like the difference in those sugars. DNA, deoxyribose, delete oxygen. So it shouldn't have that extra oxygen right there. And then RNA is just like whatever. Make sense? So phospholipids. Does anyone know another word for lipid, by the way? Simple word. Fat. Lipid means fat, essentially. Just remember, we'll talk about that in a bit later. When we talk about the phospholipid bilayer. But phospholipids, molecules that make up the bilayer of the plasma membrane and keep the membrane fluid. So it's the barrier separating the inside from the outside. It's letting some things in. It's like border control. Okay? It's like letting some things in. It's not letting some things in. It's letting things out. And we'll talk about the structure of the actual bilayer later. But so the reason it's just like little lines like this because it's just repeating. But the important thing to know is this little phosphate group at the ends. But just essentially when I see like the wiggly lines, I always just think phospholipid because it just looks very different from the other ones.
So yeah, always remember phospholipids will have a phosphate head and then lipid or fat tail. All right. Amino acids versus nucleotides versus phospholipids versus carbohydrates. So like we talked about, amino acids have that amino group, they have the carboxyl group, and they have the R group. And these are the building blocks of proteins. Where do we think we can find amino acids in the cell? What part of the cell? Cytoplasm, where else? So amino acids you could basically find them anywhere. Though most of them will be in the cytoplasm because that's where they're being made and that's where proteins have a lot of functions. But proteins work in any part of the cell. So you'll see those everywhere. Nucleotides are made up of the pentose sugar, the phosphate group, and the nitrogenous base, like we talked about. And the purpose of these nucleotides in RNA and DNA is to store and transfer genetic info. Where do we think we would find the most nucleotides in eukaryotic cells? What part of the cell?
Somebody said it?
Nucleus, right? Because that's where the DNA is stored. What about in prokaryotic cells? Where would we find nucleotides? In the cytosol, because there's no nucleus. All right, phospholipids, it has that hydrophilic glycerol head, like we talked about. Hydrophilic just means doesn't like water. I mean, it does like water. So in the anyone know how the bilayer looks? Let me draw quick on it.
I'll draw over here. Just keep up.
So basically those heads, they like water. They can interact with water. So that's why in the bilayer, the heads are on the outside and the inside because they're touching water environments. Then it has the hydrophobic fatty acid tail, which just means the tails do not like water. And that's why they arrange themselves on the inside so they can stay away from water and they're protected by the water loving heads. And that they kind of just form naturally that way. So if you were to have a bunch of phospholipids and you have water and you throw them in randomly, they would always form a bilayer like that. Where do we think we would find most of the phospholipids in a cell? Exactly. So phospholipids are mainly used in membranes. So membrane for the cell, membrane for organelles like the nucleus, anything like that. And then last one that we haven't talked about yet, carbohydrates. So carbohydrates, you'll see them off. They can be straight linear structures, but most of the time you'll see them in 5 or 6 membered rings. So if you see a ring, right away think carbohydrate. And they'll have one oxygen atom, and these are used as a source of energy most of the time. And so you can also call these saccharides or sugars. Those are the same thing as carbs. And so you can have chains of these. If you have 2 saccharides together, what do we think we would call it? It would be a disaccharide. If we have more than 2, what do we think we would call it? Polysaccharide. Exactly. All right. So I have images of all of them down here. What do we think this is right here? Carbohydrate, right? It's the ring structure with the oxygen, exactly like we talked about. Oh, and if it's a source of energy, where in the cell do we think we would find the carbohydrates? Yeah, so like the amino acids, you would find carbohydrates all over the cell. Wherever the cell needs energy, you could find them there, mainly in the cytosol. What do we think this is right here? Nucleotide, exactly. What part of the nucleotide is this? The nitrogenous base. What part is this? The pentose sugar. And what part is this? Phosphate group.
CP, think phosphate group. And does anyone know if it's, what is this? Is this DNA or RNA? RNA. Why? Because DNA is deoxy, think delete oxygen, so it shouldn't have that extra oxygen. RNA just has it.
Okay, what do we think this one is? Phospholipid, perfect, because it has the phosphate heads right here, and then it has the fatty acid tails. And what do we think this is? Amino acid, perfect. What terminal is this? What group is this? Right here? Amino. And what about this one? Carboxyl. And what part of this is different in every amino acid? R group.
And just a quick memory thing also, like hydrophobic, hydrophilic. Hydro is water, phobic, like phobia. So water hating, like it's afraid of water. And then philic means loving. So like it loves water. Yeah.
Proteins can sit, they can sit on the outside, they can sit on the inside, or they can go through the thing, through the membrane and sit on both.
Okay, stem cells. Yay, fun stuff. Okay, again, we're kind of jumping ahead here. We haven't gotten too far into the unit, but so stem cells. Stem cells are found wherever tissue regeneration can be found, so like bone marrow, liver, blood, skin, hair. And the thing that changes during its development is, again, which genes are being expressed, which is what do we call that? What's that fancy term? Differential gene expression, which means we're all made of the same stuff, but we're different because we express different things. So the only thing that's changing throughout this whole little process that you see here is just which genes are being expressed. Okay. And stem cells, because of this, have different potency. Potency. You're going to hear this term for this unit. Potency is the ability of a stem cell to differentiate into different cell types. Can somebody tell me what differentiate means? What does that mean? What's like an easier way to say that? Right. So the more a cell differentiates, the more specific it's getting. Okay. So we'll talk about the different levels of potency in a second. I also think of it as like, it's kind of like splitting down into something more and more specific. I just think specificity. I feel like that's the most helpful. The more potent you are, the more ability you have to become something specific. Okay. So let's talk about, this is my little memory trick. So there's multipotent, there's totipotent, there's pluripotent, and then there's totipotent. And I put them in this order because it's from least potent to most potent because it's in alphabetical order. So this is how it's easy for me to remember like the levels of potency in all of them without having to memorize each one. So multipotent, pluripotent, totipotent. Can somebody remind me what potency means? What does that mean? The more potent a cell is, that means they can get more what? More specific. Okay. So start on multi, go to pluri, go to toti. We'll I think I have a slide on. Okay, yeah. So multipotent, only type of stem cells adults can have, and it's limited differentiation. So it's limited in its ability to be able to do something specific. Pluripotent can differentiate into any cell except, this is the asterisk, except extra embryonic tissue. I'm sure she will ask you this question on your bottom set or on an exam. Pluripotent, I just think of it as like, it can do anything except for doing stuff that has to do with this. Extra embryonic tissue. Okay? That's the asterisk for the pluri. Yeah.
So what percentage is
I always see her use this specific term. I would stick with this just because I don't know, she's specific about things, but just having that asterisk, just knowing this information, it's like really good. And then we have totipotent. Can differentiate into any cell required for fetal development. So this is like dumb guy caveman, like can't really do much, can't really be that helpful. Pluripotent, it can do anything except for the extra embryonic, and toti is it can do anything. Okay, any questions on that? Okay, types of stem cells. So we have embryonic stem cells, which is pluripotent. So pluripotent can differentiate to anything except for stuff having to do with what? What's that term? Extra embryonic tissue. When you hear pluripotent, that's what I want you to think immediately. It can do anything except for that one. So it's the inner cell mass of the blastocyst, pluripotent cells, and they're not from the zygote. Blastocyst means, you're going to hear it. It's just a clump of cells in the early stage. Okay, that's what that means. Why is it controversial? Does anyone know why using these types of stem cells is controversial? Yeah. Yes, you have to get them from fetuses, which means you have to do damage to the fetus. Okay, because we're taking it from the blastocyst, which is a clump of cells that's in the early stage of the embryo. So essentially, you're kind of destroying the embryo a little bit to extract these stem cells. Why do we even need stem cells? Like, why are they helpful? Yeah, regeneration, right? Can anyone think of a place where it's kind of harder to get regeneration in your body if there's damage? Spine, right? So these are helpful when we need to regenerate tissue. The reason that this is controversial is because we have to kind of do some damage to the embryo / like fetus to essentially get these cells that are helpful to us. Okay, induced pluripotent stem cells. What does pluripotent do? Everything except for what? Extra embryonic tissue. Okay, iPS. These are adult cells that have been genetically reprogrammed. So we put in a little lab. We've basically made it exactly like the first guy by basically forcing it to act like this guy. So instead of having to like go and mess with the embryo and fetus stuff and like get into that whole controversy, we can just like make it and make it behave like BSE. But you know, we're making it in a lab, like it's not real, right? So it's never really the same thing. They're come from differentiated cells, they're made to de-differentiate, and it's hard to control this to make it act like the ESC. You can try your best, but it's never truly going to be the exact same thing as this. Okay, so we avoid the controversy by making it ourselves, but it's never truly going to act the same. So that's the IPS. And then the last one, adult stem cells, multipotent. Can somebody remind me, is this the least potent or the most potent? Least, right? Remember the alphabetical order? M, P, T. So it's like bone marrow stem cells, and it can be used for transplants, but it's kind of the most simple out of these three. So if she ever asks you about like, why is it controversial? She kind of talked about it in the last lecture video a little bit. That's the reason why. So people who don't want to mess with the embryo and get into like that whole argument of life thing, you can try making it in a lab like iPS cells, which we're trying to make act like the ESC cells, but it's never going to truly be the same, right? You can't recreate a whole history of a cell. You can try making it, but it's never going to be the same thing. Make sense? Again, we're kind of jumping ahead, so don't stress about it too much, but if she asks you about controversy or like, I think there might be a problem set question asking you about this.
Another thing people might use stem cells for is, say you need a new kidney and you can't find a donor. Scientists could try to use stem cells, starting with the stem cells, and turn these stem cells into a kidney. So the stem cells could differentiate into kidney cells and just regenerate and make make a whole new kidney. So that's a exciting thing that you could do with stem cells. All right. From what Anjali said, what's the difference between totipotent stem cells and pluripotent stem cells? And totipotent can be what? Anything. The way I think about it, totally anything basically. And then pluripotent, there's no really, just the alphabetical order. Think the alphabetical order thing. MPT. All right. So like we talked about, pluripotent can do everything except extra embryonic tissue, and totipotent can turn into anything.
That's it. If you guys have any questions, we're here. I hope that was helpful. We're going to send out these slides. We're going to send out the recording. Hopefully it saves normally. But please email us with any questions. We have our office hours as normal, but I hope you found that kind of helpful. And thanks for coming on a Wednesday night.
Yeah, don't be scared to stay behind for any questions or help on the problem set. We have time. I can stay for sure and help out. So yeah, thank you guys.
I'm just going to email them out. So we are basically the SIs. Like we don't get paid. So we just do everything. Yeah, pretty much. The lead TAs don't do review sessions. It's just us. We're like